Thank you, Mike. Yes, so very brief. That's my disclosure. I'm employee of Texas GI Lab. We do a microbiome testing, function testing, and I'm also the Purina Pet Care Endowed Chair in Microbiome Research, and we also get a big support from Purina for our research in the microbiome field, but that should not really affect my presentation. So we talked a lot today, and I think it's a repeated topic, and for me it's actually nice because there was so much introduction, so I don't have to go in all the details. I really want to put this somehow in perspective. And I think we need to really understand that we need to expand our basic physiology, understanding of the GI tract. When we learned in veterinary school is focus on the epithelium, on the immune system. But if you look now, and this is really a picture that my grad student Chi really did, that's really a healthy colon from a healthy cat, you can really see that's actually much more GI physiology than just epithelium. What we see usually here is a fecal sample. You can see it's actually full of bacteria. And those bacteria are metabolically active, right? So we know now that the GI tract is not really a waste pipe, it's actually a really important system where everything what enters the GI tract somehow gets metabolized by bacteria and ultimately absorbed. Then for me, we talked about this mucus layer, but this picture really shows it very drastically, that probably the mucus layer is one of the most underrated kind of safety feature in the GI tract, right? Because when you can see here in black, it really separates out all those bacteria here from the epithelium. And then we have this epithelium when we talked about barrier function. If everything is okay, I think of a very, very nicely and good functioning GI tract. And this, I think, is also a lot of confusion comes from, because all those systems are very they work together, but the GI tract and our body itself has a lot of safety features. And so, for example, this is confusion when we give antibiotics. We know now we induce severe dysbiosis. Yet it doesn't always have to be clinical science, right? Because we have this mucus layer, the gut is able to absorb many of those damages. But over time, once the system breaks down, like an ecosystem, I think that's the time which we are now realizing, once this time comes, it's actually very, very difficult to break, okay? So number one is it's very difficult to change this ecosystem. There's a lot of bacteria. It means it's not easy to change them. We know now with probiotics, we have a very mild influence. Even with diet, when you look critically at all the data, yes, diet is the most important thing to modulate, but it's mostly modulating the substrates and the interactions, but the total shift in gut microbiome is not very large. But it also means once we see a clear shift in the microbiome, it's usually a marker of something severe is happening. So that's usually a marker of something severe in the GI tract. So the most important one I want to try to talk today, that we need to look at all those components together, and they're not independent. We cannot modulate the microbiome if there's severe GI damage and vice versa. And that is really crucial, I think, because we often focus on this side or this side, but they all work together. The second important is that bacteria are really everywhere. Okay? And so this is Paula Jaretta, who was back then a PhD student. Now she's a pathologist in our lab. And she made this very beautiful picture. When you look at the colonic crypts of healthy dogs, on H&E stain, you just see a little bit of mucus. But if you stain them with silver stain, you see some bacteria showing up. With fluorescence, you see that actually the crypts are full of bacteria. And those are completely healthy dogs. And here's the challenge. What is normal? What is abnormal? If you would send a biopsy and the pathologist sees a little of bacteria, in the past, we thought immediately, oh, that must be bad. We need to give antibiotics. But interestingly, those bacteria are Helicobacter species, intestinal Helicobacter, and they actually disappear in GI disease. And so some of those colonic crypt bacteria are important because they stimulate this immune system deep in the crypts of the dog. So the fine tune is what is normal, what is abnormal. And for me, what I'm trying to say is we need to look at both the big picture and the small picture. Okay? Sometimes when we get lost in the small picture, we can overemphasize things. If we look at the big picture, we can miss some things, but I think if we learn to look at both, I think we're going to be on the right path. So how do bacteria collaborate? This is more or less a paper about the gut microbiome in cardiac disease, but I think those metabolic pathways, they are pretty much well established now that they are happening in the gut and they have different influence on all organ systems. So the gut bacteria work with direct contact, so there's this immunological role, dendritic cells, toll-like receptors, but they also metabolize many different things. And those are really dietary substrates, and that's why diet is so crucial. We can really influence with diet all those immune modulations and so on. But we also need to understand that there's many different pathways, but focusing sometimes on some of the major pathways that we already heard many times around. Here's short-chain fatty acids, because they're important for immunity, for gut modulation, and so on. We have bile acid pathway and the tryptophan pathway. Sometimes those pathways can be actually bad pathways, right? So it's a little difficult sometimes to find the right balance between gut microbiota, what is a good microbiota and bad microbiota. But looking at those pathways can really help us. The other thing is that it's very difficult, obviously, to completely describe who is there. There's many different species of bacteria. But slowly, with time, with repeated studies, we look at many studies. Here was 12 dogs, but we look at many dogs now. With time, you identify a so-called core microbiota. And what that means is really some species, they're always present in healthy animals. Okay? Of course, the little substrains and so on is more complicated, but generally, some of the basic functions are always present. And so when you look at dogs, for example, those are healthy dogs, green is present, you can see that some bacteria keep popping up, like Clostridium hydrononis, which is now renamed to Peptacetobacter hydrononis. I'm still in the old Clostridium phase. Is an important bile acid converter. It's really a core bacterium that is present in healthy animals. We see Fecalibacterium. Fecalibacterium is also like a bacterium that has been well described in human medicine like 10 years ago as one of the very first bacteria associated with health. So they have really seen that this bacterium is important for health, and usually it decreases in IBD, and it's kind of like a biomarker for IBD and so on. So we can see some of those bacteria are consistent, and they're really also consistent across different species. Important to also understand is many of those bacteria are strict anaerobes, and that means that's why we really didn't identify them until recently. So they were all of the new bacteria, and so the new concept is not necessarily looking at the bad bacteria like we used to learn. It's actually the good bacteria, and if they go away, that is probably a bad sign. So it's a really different approach. And I don't want to go much into depth because we've heard about all of this, but just a slide or 2 slides of each of some of those bacteria is to understand their role. So we know that short-chain fatty acids, we heard about them many, many times. Those are well-established markers for GI health. They come from the fiber, that's why fiber is so important. They get fermented by bacteria. There's many different bacteria that are fermenting. Fecalibacterium is one of them, for example, but for short-chain fatty acids, there's many different bacterial species and strains who can do that. But ultimately, what's important about them, that they can really bind to those G-coupled receptors and really kind of modulate immunity. So they're anti-inflammatory, they are also energy for colonocytes, and so on. In human medicine, I also know that Dr. Simpson is working on this on Fecalibacterium, but Fecalibacterium has been really studied for I think 10 to 15 years in the human field, and we also know it's an important bacterium in dogs and cats too. It's one of the core bacteria. And they've really nicely shown that it can actually work with 2 different methods. First of all, it can produce short-chain fatty acids. So it can produce butyrate, that's the metabolic pathway it is. But they also shown that it can actually some cell components itself, so there's something in the bacterium itself that can also induce interleukin-10, so it becomes anti-inflammatory. And also kind of things. So there's always 2 ways. Some bacteria can really be metabolic pathways, some of them can be truly to cell contact. And I think in Fecalibacterium, they also have shown that they can actually secrete or some immunoglobulin peptides by itself. So this is a bacterium that in human medicine now is really started to become a potential probiotic in the future. We heard also a lot of bile acids, right? So bile acids, a pathway that we usually only associate with liver disease or digestion, turns out it's an important regulator of immune homeostasis. So you can see that bile acids, they come from the liver, get converted by bacteria, and in dogs and cats, Clostridium hydrononis is the main converter of bile acids. And this ratio of those primary secondary bile acids is important because, as you can see, it's an agonist for many different receptors, even the vitamin D receptor. And ultimately, to all those receptors, we get all the different stimulations of many different pathways, like immunomodulation, but also lipid metabolism, glucose homeostasis. So this is a pathway that really has been shown to be very crucial. And this is actually also, for me, a beautiful pathway, this symbiosis, if you want to say, between the host and the microbiome, because this is a metabolite that comes from the host, from the liver, and it gets converted by bacteria into a secondary metabolite. So suddenly, it shows you how nicely how the microbiome is really part, to some degree, of our physiology. And we have shown that Paula, when she did her PhD, she actually stained the TGF-5 receptor. That's a receptor that's usually an agonist for some of those bile acids, mostly the secondary bile acids. And you can see it is extremely highly expressed in many tissues in the GI tract of dogs. So you can find them in goblet cells, in colonocytes, even in macrophages, in ganglion cells. So this shows you kind of how the receptors are really present and bile acids are really important. It also, with time, when we're looking at some of those core bacteria, we can really get the feeling how conserved those pathways are. So we're actually able to find samples, scat samples from the New Guinea Highland singing dog. I don't know if you've ever heard about this singing dog. It's the most ancient dog breed. It never has been domesticated. So that means those are really wild canines ranging in Papua New Guinea. So they're really completely separated. And as you can see, what is amazing about those, this is the reference interval of healthy dogs. And those dogs, those Highland singing dogs, have exactly the same abundance of Clostridium anones compared to our canine dogs. Even if you look at wolves, for example, raw meat diets, they also fit exactly in this abundance. And we have now samples from tigers, cheetahs, and everything, and they all fall into this range. So it's a very highly conserved species across many canines and feline species. And then also, as you can see, they're associated with secondary bile acid conversion. And then when we have, for example, antibiotics, this is really what we're discovering now when we give antibiotics, that we have those deleterious effects. So we kind of reduce the abundance of Clostridium anones, and you can see also we reduce the conversion of bile acids. If we don't do anything, some dogs come back, the bile acids come back, but you can see some animals have this persistent dysbiosis. So we can really now see that some of those functions, some of those really highly conserved functions that are crucial, they can be altered, and we can see which phenotypes altered. And we know now, at least for Clostridium anones, it's mostly antibiotics, and in chronic GI disease, and I'll come back later to that. And it's also very interesting is that this pathway by itself explains already a big shift in the gut microbiome. So we have now several studies that when you really measure the abundance of Clostridium anones by PCR, is it normal in the reference interval or decreased, and you afterwards cluster it, you can really see actually that you see clustering, meaning if you don't have your anones, you can really expect with high confidence as a huge shift in the gut microbiome. It's such an important species. And a third pathway that we already heard about, and it's so nice that we kind of repeat each other, but also brings the message home that there's really major pathways, they target some of those really same receptors, is this tryptophan indole pathway. And tryptophan indole is super exciting actually, because tryptophan by itself is an essential amino acid. We need it. Tryptophan gets converted into serotonin, so it's an important neurotransmitter. But at the same time, tryptophan gets converted by bacteria into indoles. Okay, and there's many different indole metabolites, but it's a very essential pathway. And those indoles can, again, have either direct effects, or tryptophan can have direct effects on the immune system, or via those indole metabolites, they can also stimulate the aryl hydrocarbon receptor. So again, those receptors are ubiquitous, and many different pathways can really regulate them. So this is a real abundance of it. And there's really good data out there that some of those pathways, like the tryptophan, for example, a study from 2017, they measured tryptophan by itself in serum of healthy cats versus cats with chronic GI disease. And they found that tryptophan was decreased in the serum of cats. And it also even correlated with the clinical activity index. There's also now new data now from Patrick Barker from Illinois that he also showed very nicely that those tryptophan indoles are also decreased in serum of cats with chronic GI disease and also correlate pretty well with the clinical activity index. So this tryptophan indole pathway is definitely something that is really very promising. But the question is always, why is it happening? And this is something that I want to kind of put this together, that dysbiosis or those changes, there's many reasons for that. And we have shown actually that some of the tryptophan itself is increased in the fecal samples of cats with chronic GI disease, okay? And again, for me, two different pathways. It could be one, that there's some microbial-related changes, or what we see now, and I'm going to talk about this more in detail, is this chronic malabsorption that happens in chronic GI disease. So it's a combination of chronic malabsorption, changes on the mucosal level, that sometimes may lead to losses of some of those nutrients, and ultimately all becomes like this combination of different factors. And the reason I want to talk about this a little bit is that we kind of building on Katie's and Stefan's talk, right, that we're discovering now there is a change, there's a preclinical change, something is happening, it takes some time to develop, and until we see clinical signs, usually it's already a very late stage. In human medicine, they're discovering this too. This is just a paper from this year or last year. But they really, not surprisingly, but they also show nicely that actually some vague markers of chronic GI disease are already present almost up to 8 years or 3 years before you actually have diagnosis of IBD. At the moment, they're looking at very generic markers like neutrophils and things like that, so it's not very good, but at least the concept is established. And that's, I think, important because the same thing is probably happening in dogs and cats too, and we just don't really think about it. So in humans, the model is that we have this genetic predisposition, some have it, then we have some environmental factors, which could be diet, could be other things. And then again, what we heard today, sometimes you need a second trigger, right? Something happens, could be simple gastroenteritis. Many patients probably become back to normal, but in some of them, this whole combination of dysbiosis, you get antibiotics, whatever, can slowly make those changes bad. And then we have this accumulation of inflammation. This is subclinical. At some point, there's tissue damage. We call it chronic mucosal remodeling. And once all those happens, we have a loss of GI function, and that's why it's usually persistent, right? And we're going to come back to this one that we have now, or we're working on markers that actually show that once those changes happen, once we see really severe dysbiosis, that is usually a bad sign. That's almost like a marker of chronic persistent malfunction, and ultimately potential, again, coming back to this one, that we need to understand these long-term consequences and also getting a different approach to clinical remission versus deep remission. So all of them play together. So dysbiosis does not happen by itself, okay? At least not severe dysbiosis, okay? Small changes can happen, but severe dysbiosis is always a reason for that. So once we have dysbiosis, it's very difficult to prove that dysbiosis by itself causes the disease in clinical patients, right? But in mouse models, at least, there's very strong data that dysbiosis by itself, a changed microbiome, if you induce it into susceptible host, and I think that is really important, I believe, there needs to always be some secondary components, right? Either genetic susceptibility, maybe some other damage already, then it can't really help to drive this inflammation. So this is important, I think, that we also understand it has to be several things together. Now, we heard this already from Stefan in dogs and cats, but in humans, it's well established. IBS, for example, irritable bowel syndrome, post-infectious IBS. Roughly 15% of people get it, so it's simple acute gastroenteritis. Okay, it's mostly with E. coli infection, Salmonella, Campylobacter. People go for 2 days in the toilet, then they forget about it, and a few months later, suddenly they start having those little chronic signs of IBS. Some of them normalize again, some of them actually move on to other disease phenotypes. So this is really well established. And interestingly, because we had this question today before, is it antibiotics, is it GI damage? It's probably both to some degree. So you can see that users of antibiotics during this episode actually had an increased risk compared to just having the gastroenteritis. So this is a very established reason for why you're developing those changes. Another thing is antibiotics, and especially broad-spectrum antibiotics. I think in human medicine, where they can really look at huge populations, that is important. I think we need to understand, you have to look at big epidemiology. It's clear that antibiotic, especially broad-spectrum antibiotics, are a risk factor. The very first studies have shown that it's mostly in children, but now some other studies have also shown it can happen in every single age. But I always say, you also need to look at the relative risk. And it's not really that high, right? It's still a risk factor, but ultimately it's still low. But again, saying that all of those factors together or several things have to happen together, and some of those factors we cannot control, right? We cannot really control if it happens to gastroenteritis, antibiotics. And so really risk factors, minimizing risk factors is probably at least one way for the future to move on. What we do know of dogs and cats, I think now the last 5 or 6 years we have plenty of data to show that antibiotics, not everyone, and it's interesting, mostly broad-spectrum antibiotics definitely induce severe dysbiosis. This is in healthy dogs. We did a study where we took healthy dogs. First, we put them on a hydrolyzed protein diet. You can see that dysbiosis didn't really change much. Also in the sequencing data, we didn't really see much changes in the microbiome. So diet, depending on the diet, induces some, but more minor, if minor changes. But if you give metronidazole, you can see severe dysbiosis index is super high. We stop antibiotics, and you can see it's very individual response. Some animals come back, some stay dysbiotic for at least 4 weeks. And this is really also associated, again, some of those key bacteria. You can see that's the reference interval. All animals are in a reference interval on the diet, and we give antibiotics, and some animals don't come back after 1 month. I can tell you in cats, it's even worse. Cats are extremely sensitive to almost all antibiotics. For example, Convenia, we had 1 or 2 cases on Convenia where cats, we followed this cat up for 2 months, was always normal. Then got 1 shot Convenia, 8 months was abnormal. So some of those antibiotics really have a long-lasting effect in cats. But it's not just dysbiosis, and I think for me, there's a lot of changes in the gut when you give antibiotics that potentially can damage at least the mucus layer. And so think about it, that's metronidazole, so the same animals. Here was the fecal lactate, fecal D and L lactates. And you can see it was normal during dietary transition. And when you give metronidazole, because we have this huge shift into lactic acid bacteria that could overgrow, you can see that some of those animals go up to 200 millimolar of lactate inside the lumen of the GI tract. And I don't know, but lactic acid does not seem to be a nice thing if you have so much lactic acid in your guts. And also some of those are D-lactates, which is actually a neurotoxin. So that's also kind of intriguing that you have so much D-lactate built up in the lumen of the GI tract. But again, a healthy gut with a healthy mucus layer and everything epithelium is able to withstand a lot of insults, right? Because it's built to it. But over time, we can imagine those repeated insults and so on could potentially be one of the risk factors. So ultimately, what I'm trying to say is that once we have this dysbiosis pattern, or once we have GI disease, it's not one or the other. And I think that is so crucial for me. We heard about this today already. And the second thing is there's always subsets of animals, right? One thing that for me was always interesting is we always say inflammation causes dysbiosis, and that's possible. But at least to some degree, probably even more, maybe actually malabsorption causes dysbiosis too severe, right? And the reason for that is when you look at this cartoon, that's just a cartoon that actually shows you that when we have this chronic GI disease, we're going to actually have chronic remodeling of the mucosal. We have shortening of the villi, we have less transporters absorptive, we're going to have some low-level malabsorption, okay? If you have low-level malabsorption, some of those compounds, bacteria cannot drive on them. So bacteria can actually overgrow. And then it's an overgrowth, or bacteria could produce some other substances that either cause diarrhea or they cause immune stimulation. So again, my point is not one or the other. Then we have this degraded mucus layer over time. But it's important to understand that when we look at individual patients, everyone is a little bit different. So like Katie said today, not every patient has dysbiosis. Not every patient has increased calprotectin. You know, those are very different phenotypes, and we probably need to go in this direction of staging them, because that's going to be better for us to truly optimize nutrition, optimize the therapy, and so on. And so we focused a lot recently on this kind of dysfunction of the GI tract. And so already in 2015, Julia Honefer, one of our PhD students, unfortunately never published this paper, but there was an abstract at DDW. We looked at some of those fecal metabolomics just to see what is actually altered in the GI tract in those dogs with chronic enteropathy. And what's really interesting is that, for example, you can see, and you can also see there's always a subset. It's not consistent. But one subset, for example, had increased glucose. So we are talking about here fecal glucose. So we can actually measure glucose in feces. We can measure lactates. And then you all can see a subset actually has increased fecal cholesterol. So again, this is a subset of animals, but also some of those pathways like glucose is obviously something that bacteria can drive. So we continued on this. We focused back then mostly on the glucose, on the bile acid. That was the very first kind of low-hanging fruit back then. And Paula, that was also her PhD, she looked afterwards in this expression of the ASBT receptor. That's the receptor that absorbs bile acids, okay, in the ileum. Bile acids come in the ileum, gonna be undergoing intrahepatic circulation. And what you can see nicely here, this is the ileum in a healthy dog. And you can see beautifully this long villus, right? And here in brown, that's actually the transporter. And this is kind of where the brush border is. And it's very beautiful always for me to kind of go back and say how superficial that actually is, right? How all those transporters are quite superficially. When you compare this afterwards to a dog with chronic enteropathy, you can see not just the structural change, right? It's inflammation, but you also clearly see the structural change. And there's definitely much less of those transporters. And you can see this very clearly, that the protein expression is very different in dogs with chronic enteropathy and so on. And since bile acids are so important, usually when they go, we also see that this is dysbiosis. So we have this dysbiosis that is actually mostly driven by Clostridium, okay? So this bile acid conversion, and you can also see that on average, in a subset of animals, again, we see this not conversion of bile acid. So again, it's all related to each other, right? Malabsorption, changes of those metabolites, and so on. This is again a beautiful picture from Paula that she looked at cats. So I asked her to, can you show me a cat like this? And you can see again the difference. You see this a healthy cat, normal fill and small intestine, long villi. Here in blue, already some fibrosis, but not too much. When you compare this to the lamina propria from a cat with chronic enteropathy, I think it's very clear you see the difference. Shortened villi, blunt villi, and with fibrosis, and that's important. So this malabsorption is probably more exposed in cats, and ultimately we also have this fibrosis pattern. And it also has been shown very nicely by a recent publication from Arti Katrani's lab, where they confirmed that many cats with chronic enteropathy have this already fibrosis pattern. And that's kind of understanding that at this time when we see those animals, there are already functional changes that are persistent, and all of them drive dysbiosis, all of them drive some of those metabolic changes. And we can actually see in cats, many of those markers are much more advanced when they come compared to dogs. So I think cats already have a much longer history of it. The other thing was always important is what I said before, that we're going a little bit away from the small picture, right? Going back to the big picture, because we had already today that histopathology is a tool that we can use, but it doesn't always correlate very well with clinical picture. And so Paula Jaretta with her PhD student, so she did this work where we actually looked at attached bacteria, mucosal attached bacteria, because the idea was that if you have more mucosal attached bacteria, that's maybe one reason you need antibiotics, right? That was the idea. If you have more damage, some mucosal attached bacteria, those are the animals who would actually maybe respond to antibiotics better. But it turned out there was a zero correlation. You can have here one dog has full of attached bacteria, as you can see, but it actually turned out to be steroid responsive. There was one dog with eosinophilic inflammation, it was antibiotic responsive. And a dog with neutrophilic infiltration was mostly diet responsive. So as you can see, it's a very, very almost impossible to predict from the infiltrate, from the cell culture, how animals are going to respond. And again, probably helping us that this is a destroyed system. It's an overlapping features, many things overlap, and really we kind of manage them in a way we can, but it's not really one specific disease as far we can think. And I came back to this, what drives dysbiosis? So remember, we were able to measure glucose in an untargeted approach. We now have an assay that we can measure fecal glucose with a target-validated approach, and we see actually the same thing. Some animals, again, not all of them, a subset of animals has increased fecal glucose, okay? And again, when you think about what glucose does, by itself, it could potentially cause osmotic diarrhea, right? But what's also very intriguing to me is that we have this correlation with dysbiosis. So more glucose was also more dysbiosis. And again, potentially makes sense that some of the malabsorptive patterns are a bigger driver of dysbiosis. And then you can see that there are plenty of animals who don't have this subset. And we've kind of shown this now, many studies, that we need to start understanding is not every dog has dysbiosis. They may have some, but at least the extent, the severity of dysbiosis is definitely different. And so when we look at here dogs with chronic enteropathy, clinically healthy dogs with chronic enteropathy, dogs with acute diarrhea, dogs on antibiotics, we can really see that especially when we look at the chronic enteropathy dogs, we have some dogs here in blue above 2, those are the dogs here, but we have plenty of animals that are in green, that means they're here. So there's clearly a very small, if there's a shift, it's very minor, it's a different phenotype. So there's a clear phenotype with dogs that have dysbiosis on sequencing, and it corresponds with the dysbiosis index, and so on. We also have dogs on antibiotics. But the difference between those dogs and these dogs is nowadays that, especially we can do a lot of fecal transplants. If we have antibiotic-induced dysbiosis, we have a very good chance to bring them back after fecal transplants. But usually when we see dogs that have chronic GI signs that have no antibiotic history, even fecal transplant usually does not allow them to come back. Some of them, we don't move the needle whatsoever. Some of them come back for a few weeks, but then the dysbiosis comes back again. And again, that would really, again, suggest that there is mucosal changes, mucosal remodeling. Those are the drivers. It's chronicity, and that's important to understand that dysbiosis, at least at this level, is a marker probably of chronic remodeling. So we need to kind of see it as a biomarker, but it obviously can be part of it because some animals respond really well to FMT short-term, but then they relapse again. So it's not a solution long-term because we have much more other things to do. And we're also showing now, when you look at the metabolic profiling or the metagenomic profiling between those healthy dogs and dogs with a normal dysbiosis index versus abnormal dysbiosis index, you can see very clearly there are different phenotypes, right? So dogs that have more dysbiosis have a different amino acid metabolism, vitamins, nucleotide metabolism, and so on. So clearly there's two different patterns, not just in the phylogenetic, but also in the function of the gut microbiome. And again, probably response what happens inside the host or more on the mucosal level. And so the other thing that we try to do is now better understand, because once with dysbiosis, it explains one part of a disease maybe, but we still need to have some markers that can help us actually understand what happens on a mucosal level. Inflammation is one part, but we're looking at damage. And so this is just a few things that I want to show you. Katie already showed some of the data on her dogs, but this is a different population. This is data from Federica Cagnasso that she presented at AGAF. And she looked at dogs with PLE, and we're looking at those long-chain fatty acids, and targeted assay that allows us to truly be reproducible across many studies, so you get a better feeling what's going on. And you can see really some of those long-chain fatty acids and sterols, they're very different between healthy dogs and dogs with PLE. No surprise, we can see that dogs with PLE have, in fecal sample, they lose a lot of fat. We know they respond to fat. We don't know yet if those are a predictor of low-fat diets. That's not the point of this question. It's more like a damage marker at the moment, but maybe in the future we can predict better. But in the end, it's important to understand that all of those losses, right, also cause inflammation, cause also diarrhea, and so on. So that's why this is this overlapping damage model. It's not just one or the other, and so we need to really kind of figure out what to do. Some of those markers are kind of useful, like nervonic acids. Seems to be, some of those markers are very consistent, not just in PLE, but also in chronic enteropathy without PLE, also in cats, especially in cats, those markers go up much, much higher. In fact, some nervonic acid is probably a fatty acid that is part of this gangliosides and neurons, and most likely really tells us there's more deeper damage in the GI tract. And this is actually this kind of concept we understand that clinical remission is often independent of the deep remission, deep function. Okay? So as you can see, when we have those dogs follow up 1 month, most of those dogs went into clinical remission, so clinical activity index was better. But you can see there is definitely a partial improvement after 1 month. But again, it's only partial. There's still plenty of animals who are in the middle. Some of those come down, but they're not still far away from the diseased ones. And this is a constant pattern. Here's, for example, many other studies. This is a study in Yorkshire Terrier with regular chronic enteropathy, not PLE. We can again see some of those nervonic acid and so on, but they're still coming a little bit closer, but not. But some other markers like cytosterol, for example, it's a phytosterol, is still completely abnormal after 1 year. After 1 month. And this also correlates very good with this dysbiosis that is consistent. When you look at it very carefully, yes, they come closer to healthy, but they're still not as healthy. And even the study from Philadelphia was a really great study. When you really look very carefully, even after 42 days, the bile acids are still significantly different from the healthy animals. So we get this partial improvement, so that means some animals improve, but some animals actually stay persistent, abnormal, and so on. So what does that mean ultimately? That I think we need to also long-term have follow-up samples. At the moment, most studies look at 2 weeks, 4 weeks, but I think we need to start looking at many months, 6 months, 12 months, and so on. We have a very small data sets. Those are only like, I think, 7 dogs or something that were actually able to follow up for more than 1 year. And you can see there's the healthy controls. That was at baseline, 3 weeks, 8 weeks still abnormal. After 1 year, somehow they came more normal. So this time, it takes much longer. But I can also tell you we have animals that are, for many years, stay abnormal. Okay? So this is kind of this staging disease and so on. And very soon, what is fascinating to me is that we really see a difference in acute injury versus chronic injury. Okay, so it's the same markers. When you look at dogs with AHDS, which Stefan nicely showed that they recover clinically pretty well, there are increased Clostridium perfringens, the increase of net F toxin encoding Clostridium perfringens, but they usually go away within 1 week. Okay, and you can also see those markers like arachidonic acid, nervonic acid is really very high in atopic enteritis, but day 7, day 21, it really normalizes. It all matches beautifully what we else see. So the guts, if it's around normal and you have an injury, but around it's normal, it seems to be able to recover. But once we have this chronic GI signs, usually it persists, and that's really important. And it's also very interesting to me because those dogs, and this is now the second study we have in acute hemorrhagic diarrhea, is there is a little bit of dysbiosis index. They have dysbiosis, but there is more driven by perfringens, increase in perfringens, which is self-limiting. But hyalurononis, which is actually really the one that we see in chronic GI disease, despite this really severe acute necrotic enteritis, is still normal. Again, suggesting this bacterium goes off not just by inflammation, but much more probably by this chronic dysfunction, malabsorptions, and so on. So it takes a long time for this bacterium to become abnormal. So to summarize, I think is we need to understand, we need to look at all of those things. You know, we heard today about polyphenols, and this is something that I wanted to say to Katie, is that we also know that like polyphenols, for example, have to be converted by bacteria into some bioactive molecules, right? That means some animals have dysbiosis, some don't. But in the future, we need to understand those are the genes even present, because we can throw therapies on them, but if they have a dysbiosis, they may react differently than other animals. So I think this is really crucial understanding that this subset of patients, some subset have severe dysbiosis as increased dysbiosis index. A subset has increased inflammation by calprotectin. I think malabsorption, again, is probably a level. Some of them have severe, and obviously we already saw today that also permeability. So focusing on those 4 areas can really help us, because we cannot just predict from one area what it is, but looking at 2 or 3 of those areas at the same time, it's going to be. And what is really, for me, absolutely fascinating is this acute, severe disease that still can recover. It normalizes, but we know now there's still probably some changes that at some point come back, right? So we still have this phase where we can really probably have finding markers that help us saying when they come from this acute normalization, when they start going up those markers again. And that is really, I think, the future, that we can detect this earlier. Oops. Because that really understands much better. I think we all put this in all one basket, chronic enteropathy, and it's probably the same disease. It's just different patterns, different gradients, depending where the location of the disease is, and I think that's crucial. But that's going to really allow us more tailored, patient-centric treatment, right? We optimize the treatment in the future. And again, we already talked about it, that's going to allow us much more targeted multimodal treatments and so on. And I think we also heard today, and I've really, all this data is very consistent, is early recognition is important, okay? Because some of those changes when we see are clearly persistent. With this, I want to like a lot of collaborators from many different countries. With their help, really with looking at all those samples from different areas, helps us really putting this all in perspective, and especially long-term follow-up samples are very useful, because then really can help us understanding that. With this, I'm happy to answer any questions.